Everyone, welcome back to Chapter 5. We are continuing our discussion about DNA and chromosomes, and in this particular part we're going to take a look at the cell cycle. We're going to take a look at DNA replication. Also, we're going to continue talking about how to coil up DNA so it becomes compact enough to be called chromosomes. And then towards the end, we'll take a look at histone modification and chromatin remodeling, both of which can determine if DNA is very tightly or loosely coiled. I wanted to add a quick review of the cell cycle to the chapter, and for those of us who want to learn more about the cell cycle, we'll definitely be covering it when we get to chapter 18. So this is just going to be a really quick sort of like an overview. I think it will help us with our conversation about DNA replication, which follows this particular slide. So the best way to describe the cell cycle is that it's a series of phases or steps that all the cells go through from beginning all the way to the end. The end indicating that they will go through a reproduction cycle that will allow them to produce their daughter cells, so their next generation of cells. Now, all cell cycles will start with interphase. And interphase is sort of like the day-to-day activity of the cell. This is when the cell will have a nucleus that's fully intact. Under the microscope, it will look nothing really exciting. But inside the nucleus, what we see is that the DNA is uncoiled and loose. And we see that we are full access to the genes so that you can do the day-to-day activity of the cell, including transcription and translation. So they're producing their different enzymes, their secretion. They're doing what they're supposed to be doing. So liver cells are helping with detoxification. Your lung cells are helping with gas exchange. This is just the day-to-day activity. Interphase usually takes up about 75% of the cell cycle, so the majority of the cell's life is spent in this particular phase, and it is subdivided into what we like to call G1, S, and G2 phases. G1 stands for gap phase 1. Some books will actually refer to it as growth phase 1. This is when you have a brand new cell and the cell will start increasing in size and start its metabolic function. So it will start activating the genes that it needs and it will do its functions as it's required by the body. Eventually, the cell will go into the S phase, and the S stands for synthesis. Now, the synthesis phase is going to be very important for us because this is where we're going to replicate our DNA. So this is the point where our cells go from having 46 chromosomes to doubling that number and having 92 chromosomes. So you still have one cell, but now you have double the amount of DNA that's required. After the S phase is done, you then go into G2, gap phase 2, or some like to call it growth phase 2. And G2 is all about making sure that there's no mutations, everything is ready to go, the DNA has been properly duplicated, because G2 indicates the end of interphase, and that means that we're going to start our replication phase. And that replication phase can either be mitosis or meiosis. If a cell undergoes mitosis, that means that at the end of the day, it will produce two daughter cells that are identical to the original parental cell. Identical in the sense that they will have the same number of chromosomes as the initial cell. So for us, if the cell starts off with 46 chromosomes, at the end of mitosis, it will produce two daughter cells that have 46 chromosomes. We see that our non-somatic cells, my apologies. We see that our somatic cells, I don't know what non-somatic cells are. Now I'm just making stuff up. Our somatic cells, which are our day-to-day cells, our non-reproductive cells, those are going to be using mitosis. So if your skin cell gets damaged, you use mitosis. If you want to produce more kidney cells, more liver cells, more large intestine, small intestine cells, all those type of cells, nothing to do with reproduction, they're going to do mitosis. So every time they reproduce, they produce two daughter cells that have the same amount of DNA as the original cell. So 46 chromosomes at all time. On the other hand, you can do meiosis, and meiosis is going to allow you to produce four daughter cells. And what's unique about these four daughter cells is that they will only have half the genomic material that the original parental cells has. And this is only seen in our gametes, or our sex cells. So when it's time for our body to produce eggs and sperm, it will use meiosis, because meiosis will produce cells that only have 23 copies of our chromosomes, which of course makes sense because remember, your gametes have to be haploid. You give 23, your partner gives 23, and now you have the baby that has 46. Now for our conversation, we're going to focus on mitosis. So on our illustration, we see that after the cell leaves interphase, then in order to do mitosis, it's going to have to undergo different stages. The first stage is going to be prophase, followed by metaphase, then we have anaphase, and finally we're going to have telophase. So those are the stages in order: prophase, metaphase, anaphase, and telophase. Please keep in mind that interphase is not part of mitosis. Interphase is the day-to-day activity. So in prophase, the main event is going to be that the nuclear envelope starts to break down. That means that we get rid of the nucleus. And the reason we're doing this is because the chromosomes are going to start condensing. So they're going to start coiling up, and as you can compare part 1 to part 2, notice how the chromosomes are shorter, they're more condensed, so they're coiling up. And by getting rid of the nucleus, we can now have free access to all 92 chromosomes, because remember, we doubled it in the S phase. After prophase is done, we then enter metaphase. And in metaphase, what we see happening is that the chromosomes will align right down the center of the cell. Now, some of you are looking at the illustration and you're noticing that it has this orange line like radiating outwards. This is called the spindle. This is a protein complex that will guide the chromosomes as they're transitioning from one phase to the next. So in metaphase, what's really important to point out is that the spindle and kinetochore proteins will interact with the centromere, the little middle part of your chromosome right here. They will hook themselves onto the centromere, and by hooking themselves onto the centromere, they're going to be able to separate the sister chromatids. So there's the word that we've talked about in part 1. Remember, chromatids, that means you have a duplicated side, which we do because we went from 46 to 92. So all our chromosomes have been duplicated. And what we're going to see happening is that metaphase is going to line them up in the middle, all 92 sets. And then anaphase is going to split them into 2 batches. One batch goes to the left and the other batch goes to the right. And we're hoping that if this is done correctly, then we split it right down the middle. So 46 on one end and 46 on the other hand. Now the spindle, notice how it's interacting with the chromosome, and also notice how it's contracting and it's moving it closer to either edges of the cell. So in anaphase, the sister chromatids get moved, they separate, and they move to opposite poles, 46 on one side, 46 on the other. The last phase is going to be telophase, and telophase is going to allow us for 2 important things to occur. The first thing is that we're going to reform our nucleus. So as you can see, the nucleus is coming back to us. The chromosomes are going to start uncoiling, so they become more freely accessible as they were in interphase. And then the second part is that it's going to go ahead and get rid of the spindle and start cytokinesis. Cytokinesis means splitting of the cytoplasm. So here in my illustration, notice how I have a dip inwards, and that's indicating cleavage, and the cleavage is basically showing me that my cytokinesis is about to start. Cytokinesis is an event all by itself. It happens at the tail end of telophase, and the term cytokinesis literally means splitting of the cytoplasm. And when you split the cytoplasm, you're going to end up with two daughter cells that have the same amount of chromosomes as your initial cell had. And those cells will go right back into the cell cycle, stay in interphase for the longest, and then when it's time to do mitosis, we'll move on to prophase, metaphase, anaphase, and telophase. There is a sentence that I always tell all my students that makes it a little bit easier to remember the phases in order, and it's I passed my anatomy test. So interphase, prophase, metaphase, anaphase, telophase. I passed my anatomy test. So those are the phases in order. Now, as we continue our conversation, keep an eye on the fact that in interphase, that is when the chromosomes are the least condensed. They are freely kind of like uncoiled. In the illustration, you're going to see that they kind of look like little ribbons. Whereas if you start looking at prophase, metaphase, anaphase, and telophase even, you see that the chromosomes are very tightly coiled and condensed. It all depends on if we want to just kind of have access to the genes, or if the chromosomes are being coiled up tightly because we're going to be transporting them and dividing them up. All right, so then now that we've had our little chit chat about the cell cycle, keep in mind that when it's time to duplicate your DNA, that is going to occur during interphase, during the day-to-day activity of the cell cycle. As I mentioned to you before, this is going to be the longest part of the cell cycle. The actual duplication will occur during S phase, which is your synthesis phase, and the goal is to go from 46 chromosomes to 92. And to help us with this process, we're going to rely on the spindle to move the chromosomes around and eventually start separating them. The whole purpose of doing mitosis is to start segregating our chromosomes so that once we have our 92, we can segregate them into two batches of 46, thereby producing two daughter cells that are identical in the sense of genome content and the fact that they're still going to be diploid, just like the parental cell that produced them. On the illustration that's below, you can go ahead and see that what they've done for you is they've illustrated for you interphase, and then here is your M phase standing for your metaphase. And then, of course, notice at the end of cell division that we have our daughter cells that are going to enter interphase again. Also notice on the illustration that they've shown you when you take a look at interphase that you have your nucleus fully intact and that your chromosomes are very loose, very ribbon-like, they're uncoiled, and then we need them to go ahead and duplicate. So this would be during the S phase. Notice how now there's two lines for each color of the chromosomes. Now during interphase, that's also when you're going to have all your different gene expression, because like I've mentioned many times before, it's the day-to-day activity of the particular cell. Once mitosis begins, probably the best part to visualize the chromosomes is during the metaphase stage. This is when the spindle guides the chromosomes through their centromere to line up down the center or the equator of the cell. In fact, when you look at a karyotype, you're actually looking at the chromosomes during their metaphase stage, when they're fully intact, they're tightly coiled up, and they are lined up right down the middle of the cell, making them easier to align with their homologous pairs. And then as mitosis comes to the end, we see that we will go ahead and reform the nucleus. And as the cells, the new daughter cells enter interphase, we see that their chromosomes will uncoil again, allowing them to once again start their gene expression. And when it's time for the daughter cells to go into S phase, they will also duplicate their chromosomes as well. In order for the duplication process to be successful from a chromosome standpoint, we're going to have to focus on three regions of the chromosome. We have a replication origin, we have a telomere, well, actually we have two of them on each chromosome. Those are going to be the end caps. And then towards the middle of the chromosome, you're going to have the centromere region. That is where your kinetochore proteins from your spindle are going to be making physical contact so they can move the chromosomes towards the equator of the cell and eventually split them into two separate batches. The replication origin, as the name indicates, that is where the DNA duplication is actually going to be initiated and where it's going to start. Let's go ahead and skip over to the next slide so we can take a look at an illustration of these three structures. Alrighty, so here is my DNA sequence, and what we're going to do is we're going to start taking a look at it in interphase. So as you can see in interphase, what we see happening is that we have our three regions. At the end part, we have our telomeres. So each chromosome has two telomeres, one on top, one at the bottom. Consider it a little cap. You have multiple replications origins, here indicated by yellow. And the replication origin is basically the location where we're going to see our first signs of DNA replication. And that's often indicated by the fact that the helix will start separating itself so that we can get access to the bases. we can get access to the sequence, so the A, the T, the G, and the C. You can have multiple origins, and usually when they start replicating, they will do so in a bidirectional pattern, meaning that they'll reproduce or they'll replicate both upwards and downwards simultaneously. Okay, so they reproduce or they replicate, my apologies, bidirectionally. So they go both ways, up and down. And then as you can see right here, we have our centromere, and I said something before that wasn't completely correct. I said the centromere is at the center of the chromosome, and that's not true. It is located either towards the center or more towards the edges. There's quite a bit of variation at the centromere location, but the centromere is more important, not where it's located, but for the fact that the mitotic spindle will be able to make physical contact with the chromosome at that particular location. And you generally just have one centromere. So we'll have one centromere, multiple origins, and then you'll have 2 telomeres that we're looking at in a typical chromosome. As you follow the sequence of events from left to right, it's basically showing you that somewhere in interphase you will start your replication pattern. That's of course in phase S for synthesis. That will allow you to have your duplicated chromosomes, which means that when I do my metaphase, it will allow me to align them right down the middle of the cell and my spindle to interact with the centromere, so I can separate them back into 2 separate batches of 46 chromosomes. And all the daughter cells at the end of the cell division will automatically enter interphase, because that's the first step of the cell cycle, and they'll stay there for the majority of their life during their day-to-day activity. The image that's all the way on your right-hand side is just kind of showing you what a chromosome looks like under an electron microscope, and then next to it is a little illustrated example just showing you the fact that you're looking at a duplicated chromosome. And in case you were wondering, these 2 are sister chromatids. See right here? One of them is called a chromatin, because that's the coiled up DNA. Remember, we talked about that in our last part, the difference between chromatin and chromatid. So because it's duplicated, it's a chromatid. Alrighty. Another thing that's really interesting, I find, is that through the use of fluorescent staining and, of course, the use of an electron microscope, scientists have figured out that there are very distinctive regions that are dedicated to each and every single one of our chromosomes. And part of that comes from the fact, and we'll talk more about this in a slide that's coming up, is that as the chromosome or as the DNA starts its compacting method and starts to condense more and more, there will come a time where the chromosomes will start to interact with the membrane of the nuclear envelope itself. That particular region is called the nuclear lamina. The nuclear lamina is basically a little protein meshwork that you'll find within the inner lining of the nuclear envelope. And what we see happening is that the chromosomes will interact with certain proteins that are found in this meshwork. And as they start interacting, they become entangled and attached to the nuclear envelope. And they'll start forming what we call chromosome territories, meaning that each chromosome will have its own dedicated space within the nucleus. So go ahead and take a look at the picture that's labeled B. And you can see that what they've done is they've highlighted some of the chromosomes. So in red, you can see chromosome 11, blue is for 5, and 3 is shown as green. Each of these chromosomes has their own distinctive chromosome territories within the nucleus. And also notice how the homologous chromosomes do not necessarily need to be aligned next to each other in the same territory. So notice how 11, 5, and 3, each of them are split between obviously their paternal and the maternal contribution. So I think that's definitely something that I wanted to point out to you, the fact that the chromosomes will have their individual territories within the nucleus due to their interaction with the nuclear lamina. Now, obviously, there are many steps to go before you can actually go from naked DNA to chromosomes. And this I sort of highlighted for you when we were chit-chatting in part 1. But I want to kind of go back and take a look at a little bit more detail. And one of the things I wanted to look at was the formation of nucleosomes. A nucleosome is basically DNA plus histones. So what we see happening is we can take our naked strand of DNA and we can find proteins called histones. And these histones will go ahead and they'll work in what we like to call an octamer core, meaning that there will be 8 of them that'll be stacked together. Let me see if I can draw it real quick. So you'll have 8 of them. These are 4 in the front, 4 in the back. So you'll have an octamer, you'll have 8 histones stacked together. And then what we see happening is that the DNA will come around about 150 base pairs. To be exact, it's about 147 nucleotides will come around. That's the length of DNA, and it will wrap itself around the histones. Now, obviously, my drawing isn't the best, but what you can do is take a look at the picture right over here. The yellow is meant to represent the little octamer core. So those are your 8 histone proteins that are kind of clumped together. And then in red, we see the naked piece of DNA that starts to wrap itself around. Now, this particular complex, so the core with the DNA wrapped around, that is what we call a nucleosome. And the nucleosomes will go ahead and start stacking on top of each other. And that is then what's going to eventually give rise to chromatin. So chromatin is nothing but nucleosomes just stacked on top of each other. And in order for the nucleosomes to do that, we see that they need to rely on those octamer cores, which basically are composed of histone 2A, histone 2B, histone 3, and histone 4. Structurally, they're just slightly different. So these 4, 2A, 2B, 3, and 4, each of them will have 2, making it a grand total of 8 histones within the octamer core, hence the name. Octamer means 8. These histones are highly conservative proteins, meaning that we come across them in a lot of eukaryotic organisms, and they're very similar in size and function across the way. So they're highly conserved. The other thing is that the cell will have massive amounts of histone. One research paper actually indicated that there could be over 60 million histones per individual cell, and that's because it is so essential to compacting the DNA and initiating the process of forming chromatin, as well as eventually leading its way over to the chromosome formation. But I'm getting a little bit ahead of myself. If you go ahead and take a look at the picture that's at the bottom right here, we can see that as the nucleosomes become formed, they sort of look like little beads on a string. And you're also going to notice that you have a little line right here, right? A little area from one nucleosome to the next. That is going to be called linker DNA. And linker DNA is basically just a piece of the DNA that is not interacting with the octamer core. Instead, it's going to be interacting with another histone called H1. H1 is not a core histone, but it's a linker core because it's going to be able to regulate the length of DNA between the individual nucleosomes. Now, the picture that's been inserted right here, figure 5-20, basically talks about the fact that in a laboratory setting, we can easily uncoil the DNA and its nucleosomes by exposing it to a nuclease enzyme. The nuclease enzyme will go ahead and start digesting the linker DNA. You can see the pieces start to fall apart. And as that happens, what we see is that the core then becomes separated. So the individual nucleosomes become separated. And usually you'll have a kit that you can purchase, but you're basically would just alter the environment of the proteins. So salt will do it. So in this case, it says it was those exposed to high concentrations of salt. And that will allow, obviously, the octamer to fall apart into their individual pieces. So you can see the representation of the paired 2A, 2B, 3, and 4 histone protein. And then, of course, the DNA itself, you can see right here, 147 nucleotide pair length. then becomes uncoiled and becomes easily accessible due to the fact that it's no longer interacting with the protein. I also want to kind of mention to you that one of the reasons why the histones are so successful is due to the fact that they naturally have a positive charge, so they're able to attract the DNA and hold onto it very tightly, obviously due to the fact that DNA itself has a negative charge because of the formation of the phosphodiester bonds, which is something we discussed in part 1. Here is just another look at the core histones, and this is also the part where I want to show you, or I want to kind of point out to you that, and I love this picture by the way, isn't this gorgeous? So this one shows the picture of the core histones, the octamer, allowing the DNA helix, which is indicated by this gray area right here, to wrap itself around, thereby compacting it down. And as you can see, we have a little tail that sticks out. That's going to be important in a few seconds. And then to make sure that we keep a distance from one nucleosome to the next, you're going to have the linker histone H1 come into play. Now, the linker is going to be important because it turns out that H1, as well as other non-histone proteins, will go ahead and interact with the nucleosomes. And what we see happening is to increase its compaction, these non-histone proteins will take our little chromatin and they will start forming loops. So they're condensing it down even further, taking it from a long strand of beads to like a loop formation. And as they're forming the loops, they're allowing the condensation to continue on and for it to become even more compact. Here is a picture that I inserted. This one comes from a McGraw-Hill book, but I liked it because it definitely shows you how your H1 histone is fully interacting with the linker DNA, which is obviously the little cord of DNA that we have from one nucleosome to the next. This larger entity right here, that one is representing the histone octamer. So that is where your 8 other histone proteins are. So the 2A, the 2B, the 3, and the 4. So this is a little bit of overkill. I know we've discussed it on the previous 2 slides, but altogether there are 5 different types of histone. 2A, 2B, 3, and 4 are part of what we like to call the core histones. 2 of each, so they make an octamer, and then H1 is going to be a linker histone, and that's because it's going to be binding to the linker DNA. So keep in mind that in addition of H1, the linker, we will also have other non-histone proteins that will start interacting with the linker region, and this will then greatly affect whether we are tightly coiled, meaning there's only a slight piece of linker DNA and the nucleosomes are very close together, or if it's loosely coiled, meaning that there tends to be a little bit more of linker DNA and the nucleosomes are not as tightly compacted as we would expect them to be. All of this will depend on whether we would like the gene expression to be active or not. So here we have this beautiful illustration that basically shows us all the different steps or phases that DNA has to go through to go from naked DNA to forming a chromosome. On top, we can see that we have our double helix. So this is how we're used to seeing DNA when we look at it in like our book and our notes and whatnot. We see the double helix, we see the phosphate diester bond. We're able to discuss the A with the T and the G with the C. And then if it's time to start coiling it up, we see that the next step is to go ahead and find the histone proteins so that it can make a nucleosome. Here's my little nucleosome. And the nucleosomes, when they start stacking on top of each other, they will go ahead and be condensed to the part that we call it chromatin. So chromatin is coiled up DNA. It's basically coiled up nucleosomes, which of course are histones plus about 150 base pairs of DNA. The next step is that after the chromatin, we're going to form the 30 nm nanometer fiber. And that basically means that we're going to start manipulating the linker DNA and compacting it closer and closer together so that it goes ahead and measures around 30 nanometers right over here. Now, this is a particular shape that the DNA takes on, so it's part of its compaction level, and it turns out that after the fiber is formed, we now see that the chromatin will start interacting with the lamina of the nuclear envelope. Remember how we talked about the different regions or territories that each chromosome has? So this is the part where we see it interacting with the mesh network that we find within the nuclear envelope, and it's going to allow the chromosomes, or I should say the 30 nm fiber, to start forming these loops. And as you can see right here, the loops will further condense down the DNA, and we're going to call this radial loops to indicate that it's interacting with the nuclear envelope. And as it's condensing down, it will get down to the point where we can go ahead and we can call it a chromosome. And by the time we get to the chromosome, we've had about a 10,000-fold condensation. Look at that. Isn't that crazy? And that is then how we're able to house massive amount of DNA in the small nucleus of our little cells. So we went from double helix to nucleosome to chromatin to the 30 nm fiber to the radial loops, and now we're able to form our chromosomes. That was a lot of work, but as you can see, it's totally worth the effort because you're able to condense a large amount of DNA into a small place. Now, it turns out that once all this is done, the chromosome will not always stay in its regular shape. And part of us already know that because we talked about the fact that DNA is rather loose when it's in interphase and will only start condensing when it's ready to start its prophase, metaphase, anaphase, and telophase step of mitosis. So we already know that the chromosome can uncoil and coil back up. But one thing we also have to discuss is that within the chromosome itself, there will be sections where the DNA is very tightly coiled up, and there will be other sections where it's easily available and not as tightly coiled. And that will all have to do with gene expressions. Remember that within your DNA strand, you have a genomic region, and if those genes need to be available, that section of DNA cannot be tightly coiled because we need to have easy access to do our transcription and translation. Other ends of the chromosomes that are not being used by the gene, so maybe the gene is silent or maybe it's a non-genomic section, those sections will be tightly coiled because there's no use of exposing them to any additional enzymes or whatnot. They are literally just taking up space and maintaining the size of the chromosomes. So we have to have a way to kind of manipulate the level of DNA compaction within the chromosome. And that is what the last part of the chapter is going to take a look at. In order for us to regulate or modify our chromosome structure, we basically see that we can do what we call histone modification, and we can also use chromatin remodeling complexes. And the collection of these two, the histone modification and the chromatin remodeling complexes, those are going to allow us to have the chromosome either be in what we like to call heterochromatin, which means it's very compact and tightly coiled, or euchromatin, which means that it's still compact, but it's not as dense, it's loose, meaning easy access for the genes that we're trying to do transcription and translation on, so the ones that are active within that particular cell. So let's take a look at histone modification and then chromatin remodeling complexes. Now, as you see from the name, histone modification means that what we're going to do is we're going to go over to the tail end of the histone protein, and we're going to either add or remove and do a reversible chemical modification. And as you can see right here on the slide, I wrote for you that on the tail end of the histone, as you can see, the tails are just kind of hanging out. They're extending outside of the octamer. On the tail end, we can do methylation, phosphorylation, acetylation. And when we do this, that means that we're either using covalent attractions to either add or remove. Are we adding a phosphate group or removing it? Are we adding some methylation or are we removing it? So you're starting to manipulate the chemical structure that you find at the tail end of the particular histone. The reason we're doing that is because it turns out by modifying the tail end, you can dictate the stretch of chromatin that the non-histone proteins have access to. And by attracting different non-histone proteins, we can basically alter the compaction rate of the nucleosome. So will they have a lot of linker DNA, or will they only have a short piece of linker DNA because they're being tightly coiled? This plays an extremely important role in gene expression and gene silencing, because in our previous slide I said to you that if we have a particular gene that needs to be expressed, so we need to do transcription and translation on them, we need that particular piece of DNA to be slightly uncoiled and easily accessible. Whereas if there's a particular gene that we're not using that we want to silence, we can coil that up tightly, almost like we're putting it in storage. So thereby, we're not encouraging transcription and translation. So for instance, we can go ahead and take a look at the illustration. That's figure 5-25, and it shows us a little modification on the histone tail, and we're looking at H3, the tail of it. And what we see happening is that we can go ahead and see if we're concentrating on the K9 position, depending on which chemical modification is done, it will determine if that gene is available or unavailable. So for instance, in the top spot, you can see that methylation has occurred. They went ahead and they added that chemical component, and by adding the methylation, we see that it allows the DNA, or I should say, it encourages the histone to coil up that particular piece of DNA even tighter with the use of other non-histone proteins. And because it's tightly coiled, we call that heterochromatin. I'll show you another picture in a little bit, but because it's so tightly coiled, we also see that there's gene silencing. So there's no access to that particular piece of the DNA. What if we take the methylation off and instead we're going to do acetation? So you can see right here, it's being added. So maybe we are we're using transferase, which is a particular enzyme that you can use to move the acetyl group. So if you're doing your histone acetylide transversase, we see that it's added over to our particular location at the tail. It makes the tail more attracted to other non-histone proteins. So in this case, we see that it loosens the linker gene, or the linker DNA, which allows for that particular gene to become more accessible. So now we are able to do full-on gene expression of that particular section of our DNA. Now, along with histone modification, we also see that hand in hand, you'll have chromatin remodeling complexes that will assist on determining how tightly the nucleosomes are connected or how loosely they are from each other. Now, the chromatin remodeling complexes, these are ATP-driven large protein machines that basically, as you can see in the illustration here in green, they will wrap themselves around the histones and fully interact with the compaction level. And since it's ATP dependent, notice how they do ATP hydrolysis, so they can free up that chemical energy and they can utilize that to go ahead and do some sliding down the nucleosomes. And by doing so, that means that they can go ahead and take a chromatin section that was very condensed, and they can start decondensing it. So they can start uncoiling it. And the more DNA they make available, the easier it is for genes in these particular regions to be expressed. We also see that in extreme cases, it can decondense the chromatin so much to the point that it will actually kick the histone out of that particular section of DNA, freeing that entire 147 nucleotide sequence that was wrapped around the octamer. Now, because the histone modification and the chromatin remodeling complexes work hand in hand together, it kind of explains why DNA oftentimes can make a very rapid transition from condensed to loose. So we can quickly alter which genes are expressed and which ones are not. This also explains why you have the same amount or the same type of DNA in all of your cells, yet each cell will have its own unique function. So for instance, your skin cells have the same 46 chromosomes, but they have a completely different function than your stomach cells or your liver cells or your kidney cells, because we see that through the use of these modifications, we're able, or your DNA is able to control which genes are activated and which ones are silenced. So by manipulating those genes, you can also manipulate the overall function of the cell that you are looking at. And because you're able to uncoil and condense back the chromosomes by the histone and the chromatin level, we can then go ahead and see that most chromosomes will have sections that are euchromatin and some sections that will be heterochromatin. Heterochromatin, as I mentioned to you before, is the highest level of compaction. So this is when the chromosomes are very tightly coiled. This is going to correlate with sections like the telomeres, the centromeres, and also areas that are silenced by the genes. So here you have a gene that you don't need, so we're going to silence it. So what we're going to do is we're going to tightly coil up that section. And usually, guys, just to let you know, about 10% of the chromosome will be in heterochromatin mode. And that basically also indicates the fact that cells don't have all their genes activated all the time. Most research papers will tell you that on average, a regular cell will only activate about 50% of the genes it has access to. And even when it activates the genes, it does it at a very low level. So it just seems like a little bit goes a long way. So within a chromosome, there really is no uniform pact. There will be some areas that will be highly compact, heterochromatin, and then obviously there will be some areas that we need the genes to be actively expressed and easily accessible, and those are going to be in euchromatin. So you can see it here indicated in the blue and green sections. Notice how that is not as tightly coiled. So the different proteins can have free access to the DNA that needs to be expressed. I want to give you a really nice example of DNA compaction and being the ability to kind of silence a gene by putting it in the heterochromatin. So let's go ahead and look on our next slide, and let's talk about this process called X-inactivation. All right, so what we see happening, and this happens in different species, but we'll talk about it for mammals. In the female species for mammals, because we inherit two X's versus the male, or I should say the females inherit two X's, and the males inherit one X and one Y, there seems to be a discrepancy in the dosage of how many genes are expressed between males and females. Because females have two X's, that would technically mean that they are able to express twice as many genes on the X chromosomes. Our body does not require two X's for expression. We only need one. So what we see happening is very early on in embryonic state, at random, one of the X chromosomes will become inactivated. And the way it's going to get inactivated is that the chromosome is going to be coiled up so tightly that it's going to go ahead and form what we like to call a bar body. Here, a bar body. And the bar body gets its name from the staining process that's done to visualize it. Because it's so highly condensed, it will soak up a lot of stain, and it will actually appear as a dark spot under the microscope. So here in my little illustration, notice how in the female nucleus you have your little bar body. In the male nucleus you don't, and that's because males only have one X, so that one will be fully activated at all times. Females, we will have our inactivated X, and then here you can see on the illustrated example, this is the X chromosome that's going to remain active. Two important things to point out. One, it happens very early on in embryo, and the cell will remember which one of the X chromosomes is at randomly inactivated, and as it goes through its mitosis step, all those cells that follow will have the same X chromosome inactivated. So the other thing that you want to keep in mind is that it's at random. So you can't really control if it's going to be the X that you have from the paternal, that's what the little P stands for, or the maternal lineage. So you don't know if dad's contribution or mom's contribution is going to be silenced. So that's why on this illustration right here, it says inactivation of a randomly selected X chromosome. And over here on my left-hand side, I see that dad's chromosome has been inactivated. So we are expressing the one that you received from mom. On the other side, we see that mom's chromosome has been inactivated. So we are expressing our paternal heritage from our dad. And because it's randomly inactivated and because of the memory that the cell maintains, we see that as the cells continue to divide, all of the daughter cells, all of the descendants, will have the same inactivation pattern. Now, a really good example of this at work is when we take a look at calico cats. So let's go ahead and let me show you a picture of those. All right, isn't that a beautiful cat? Look at that. So calico cat is a cat that's distinguished for the fact that it has these beautiful patches of orange and black fur. They are only females that get the name, and the reason they have these beautiful patches is because their fur color is actually determined by the X chromosome. One gene will code for the black fur, and the other version of the allele will code for the orange fur. Now, depending on which X is inactivated will determine what color will show through, and the patches that you see happening, so for instance, right here you see a beautiful patch of black. This basically indicates a collection of cells. You can think of them as descendant cells or daughter cells that all originated from that one cell that had randomly inactivated the orange color, which allowed for the black color to seep through. Whereas the orange patch, those are all descendant cells from a very early embryonic cell that at randomly inactivated the X that coded for black fur and allowed the X that coded for orange fur to shine through. Isn't that beautiful? And like I said to you, this happens very early on in embryonic development. You can't really control it, so each pattern will be slightly different from cat to cat. Another example, and most likely you'll talk more about this in your genetics classes, but obviously if you have a question about it, I would love to chit-chat about it, is the fact that if we're looking at certain genetic disorders, if it's carried on the X chromosome, we see that it tends to affect the male species more than the female species. And the reason for that is because males only get one X chromosome. So if there's an abnormality, they automatically have the disease, whereas females, depending obviously on the disease, if it affects one X chromosome, they do have the backup of a second chromosome, which means that sometimes they can be classified as carriers, meaning that they have the marker for the disease, but they don't have any of the symptoms because they have that backup X chromosome to kind of heal up the process. So for instance, in color blindness, color determination is found on a gene that's on our X chromosome. It is much more common for a male to be colorblind than for a female to be colorblind. Why? Well, because the male only has one X chromosome. So if he has a mutation in that colorblind gene, he automatically has the disease. Whereas for females, if she has a mutation in that in the color determining gene, we see that that X has the ability to become inactivated, and she has her backup X chromosome that will allow us to see colors like the wild type, so like normal. Is it possible for a female to become colorblind? Yes, absolutely. But in order for that to happen, we need to see that both of her X's have a mutation on it. So we need to have two mutations, one on each X, and that's the only way that a female can become colorblind. This would also indicate that the father of the female would be colorblind, right? And he's passing on that gene, and then the mother can either be a carrier, meaning that she has one mutated X, or she could be colorblind herself, meaning that she has both mutated X's available. So that's genetics at play. So, all right, I'm going to stop talking over here. I hope you guys enjoyed Chapter 5 as much as I enjoyed chit-chatting about it. As always, if you have any questions, comments, or concern, please feel free to send me an email, and I'll be more than happy to elaborate. All right, everyone, enjoy the rest of your day.